Hey, this is Joel Duff. Let's talk about a paper that was just published yesterday. This paper discusses a pernicious problem in environmental DNA research. Environmental DNA is the extraction of DNA straight from the environment, whether it's from water, soil, or right out of the air. Collecting DNA from the air and sequencing it, and then asking what DNA is in the air. So what's the problem? What's this pernicious problem? The pernicious problem is that virtually all captures of DNA from the environment include human samples, right? Endogenous human genomic samples, or in this case, they're calling it inadvertent human genomic bycatch, all right? From intentional capture, right? We're intentionally trying to capture information about the environment. That would include the different types of organisms that live in a particular environment. And one way of finding out what organisms live in an environment is to simply take a snapshot of all the DNA that's present in that environment, trace back the origins or trace back the origins of that DNA to what organism was that piece of DNA associated with when that organism was alive. Right? And you can do that by sequencing massive numbers of little bits of sequence from the environment and then asking, what is this sequence most like? Is it most like a bird or a mammal or a bacteria? Right? That would be large groupings. But if you have enough sequence, you can hone it right down. You can drill down to the point where you get to, oh, this is this genus, or this is within this is a particular species. And if you had enough prior knowledge of individuals, for example, individual human beings, you might get enough sequence information to be able to say that individual was present in this particular environment. And that's the brave new world we've entered, right? With environmental DNA, especially with how sensitive our ability is to be able to, how good we're getting at extracting DNA from our environment and realizing that our DNA that we live behind as I am breathing, as I am sloughing off cells, as I walk through the sand on the beach, as we're going to see in this particular paper, I am leaving my DNA behind, which for days, weeks, months, potentially for years, potentially for dozens of years, hundreds of years, and even in some cases, thousands and thousands of years, somebody can find evidence that I once was at that particular place and point in time because I have left my genomic imprint on that particular location. All right, so this paper comes to us from Nature, Ecology, and Evolution. It's an open access paper. One of the reasons I feel like I can show it to you, even though it's published yesterday. We see that it's by the David Duffy Lab at the University of Florida. And a bunch of colleagues, and what they are mostly interested, let me scroll down here and you can see this is, many of these colleagues are from the Whitney Laboratory of Marine Science and Sea Turtle Hospital. And they were particularly interested in green turtles and identifying locations, identifying green turtles and their populations and so forth. All right, but that's not what this paper is about. It's sort of like, we're studying green turtles, and what we realize is that in all of our environmental sampling we're doing in order to track turtle populations, we'll always have human DNA, right? And the question becomes, what do you do with that human DNA, right? What do you do with all of this human bycatch, right? You're catching samples of humans in your samples as you're extracting DNA from the environment. So now we get into a variety of different thorny topics, right? Because you have things like, in order to publish a paper that involves doing DNA sequencing, there's essentially a worldwide agreement that to publish research, you need to publish the DNA sequences that you use to do that research, right? So that anyone else can check your results. And so there are large databases. National Institutes of Health has a huge database where I've sequenced many different organisms, genes from different organisms, and in order to publish my results, I submit my sequences to that database and I get an accession number, and I put that accession number in a paper. Thereby, if you read my paper, there's the accession number. You can go look up that particular sequence and see that sequence and analyze it for yourself and see whether I've made any mistakes in my particular analysis. So now, if you're doing an environmental DNA study, and I've done these environmental DNA things, it's one of the reasons I'm talking about this. All right, and you've captured thousands, tens of thousands, hundreds of thousands, or today, billions, in some cases, hundreds of billions of sequences, right, from the environment. Short fragments all the way to even fairly large fragments, up to millions of base pairs each. You take all those fragments and then you would submit them to a large, it's a very large database, and you submit that as a file like, here's all my environmental DNA sample. Well, by doing that, if there's no, the question becomes, what do you do with all the human bycatch, right? That human DNA is part of the environment, it's part of the environmental DNA sample, and as we're going to see in these figures, it's hard to sample really most places on this earth without capturing human DNA at the same time. There's also a secondary issue of our instruments we're using and the fact that humans are extracting DNA. We can contaminate our own samples with our own DNA. That problem actually isn't quite as bad as the actual just native DNA in the environment that we're extracting. So we need to submit all your human DNA to this database, right? Potentially that includes signatures, right, of particular individuals. Little short fragments, of, what do you mean by short, Joel? Short would be 15, 100, 200, 500 base pairs of DNA. Typically 500 base pairs of DNA isn't going to have enough unique signatures to be able to identify you, right? 500 base pairs is probably enough, is going to be enough to identify that this is human DNA in many cases, right? We have enough specific base pairs of a particular gene that gets sequenced that it would be recognized that is a member of the human population versus some other mammalian population. But once you get up into looking at tens of thousands of base pairs, right, fragments that are 1 million base pairs long, there's probably enough unique signatures in the particular set of sequence that individual has that if that person's DNA were in a database, like I've had my genome sequenced and it was available for someone to search, I could be connected to that environmental DNA. And so now this is going to get us to all the different applications, potential applications, both good and bad, of environmental DNA, in that we might be able to use it for, tracking down criminals, all right? But it could also be used for tracking down anyone for any other nefarious purpose as well. Let me read the abstract to this paper. I'm going to explain a little bit about a tiny bit of the methods of this paper, because you might be interested like how we get this environmental DNA and like the cost of it, because I think it's important to know how easy this is to do. And this is becoming easier every single day. And therefore, that's why the applications of environmental DNA are expanding constantly. And you should, if you pay attention at all to science news, you'll see that you hear a new thing about environmental DNA seemingly every single day. The field of environmental DNA is advancing rapidly. Just said that. Yet human environmental DNA applications remain underutilized and underconsidered. We do use environmental DNA for a lot of stuff with humans. Just think back to the recent pandemic, and we sample wastewater and stuff like that in order to find out how many people are shedding viruses. We've discovered that people have polio in certain places because there's individuals shedding that polio sequence. Now, that's not directly identifying humans, but it's identifying products from humans. Right? Broader adoption of environmental DNA analysis will produce many well-recognized benefits for pathogen surveillance, biodiversity monitoring. We use environmental DNA to identify, we can sample one site multiple times over years and find out how many different types of organisms are living in there, what percentage of organisms, of each different type of organism is living in a particular soil sample or in a tree or, in the surrounding, in the water. And then you can do that sample over and over and over again over many, many years, and you can track changes in diversity, changes in even population sizes potentially. We can track endangered and invasive species. How do you know that this particular endangered species is here? Do you have to set up traps in order to collect this particular small animal, or can you simply just sample the environment, sequence a whole bunch of DNA, and like, oh, the DNA of that particular animal is present in this environment, so you know that that animal has invaded that environment. And then, of course, population genetics. We can get a really big snapshot of the diversity of even populations of organisms. So that includes populations of humans, all right, in areas. Here we show that deep sequencing-based environmental DNA approaches capture genomic information from humans. Deep sequencing-based eDNA is just referring to, like, we're looking at more than hundreds of sequences. We're usually looking at tens of millions to billions of sequences from an environmental sample that we've taken. I'll talk a little bit about these samples in a few moments. And in those samples, we capture genomic information from human beings, just as easily as we do from the intended target species. We may have something we're researching, right? We're interested in either a particular organism in the environment, or in my case, I was interested in microplankton, working with a colleague. That was basically his research. He traveled to the Arctic and took samples of water samples from different depths, different salinities in the Arctic Ocean. And then I took those water samples and I extracted DNA from them. And then I told him, here's what organisms are living in those samples. And so we're capturing a population profile of the different kinds of organisms in different water bodies and different places in the Arctic. So basically taking a transect and you're seeing, like, at different depths, different salinities, which usually is because of mixing of the Arctic Ocean with other ocean waters. What different populations of organisms are living in those different waters, and how are those affected seasonality, and then of course in terms of climate change, how much is happening over many different years, because that's also what environmental DNA is allowing us to do really easily now, is to track with much greater precision the balance of populations and species composition over months, over years, and eventually over decades, and we'll get a much greater picture of the changing, the way life changes on Earth. Okay, but let's progress through this abstract. We term this phenomena human genetic bycatch, HGB. Additionally, high-quality human environmental DNA could be intentionally recovered from environmental substrates, water, sand, and air. Yes, we could, if we are specifically looking for humans, right, and their presence in the air, right, come into my home, take an air sample, collect the DNA from that air sample, right, and then sequence it, and you're probably getting enough sequence that you would be able to tell if once databases become large enough, or if sequence that happens to already be in the database of some of individual family members, then they'll be able to say, yes, those particular individuals are in that house. Holding promise for beneficial medical, forensic, and environmental applications. They have a chart in this paper, which I'm going to show you, which is like, here's a list of things that are going to be positive benefits of this technology. However, this also raises ethical dilemmas from consent. Did I give you my consent to sequence my DNA? What rights do you have to your own genome once it's sort of left your body, right? I'm waving my arms around. There's cells falling off right now, and they're floating in the air, right? They're going to waft out the window, and if somebody takes an environmental sample out there, they potentially could sequence my entire genome. At least they're going to sequence large portions of my genome, and therefore have my data in a database, my sequence data in a database. All right, so yes, that raises ethical dilemmas. Is that my DNA now, or do I not have the rights to that information afterwards? Do I need to consent to that? Privacy and surveillance, data ownership. Who owns this data? Right? Stuff goes into databases. Who owns it? Who guards the gates of who can see it and who can use that particular data? Right now we have massive public databases because a lot of this research is done with public dollars, your tax dollars, and you have the right to see the types of information that your tax dollars are generating. But at what point do privacy issues come into play? This requires further consideration, potentially novel regulation. Yes, there might need to be regulation. Should there be regulation on how police can use these DNA databases? We've already had many famous cases of criminals that have been caught because their DNA samples have been found at locations. And although there was sort of a private database of DNA type stuff that has been used before, now they're simply just submitting DNA samples as if they're like, hey, I'm submitting it to an ancestry service, right? And here's all these people in the Ancestry Service, and they use the Ancestry Service to identify what this DNA is related to. And maybe somebody in that ancestry, maybe somebody who submitted their DNA to the Ancestry Service is related to that DNA sample, or may actually be the same person as that DNA sample, in which case they're the criminal, right? But even if you get close to the criminal in the sense of, hey, there's a family member, that's a clue, all right? Oh, I need to look at people that are related to this individual, and that's where I'm going to find my criminal. Right? And so this has already been done. There's been lots of controversy generated by that in terms of the privacy of the individuals who had their DNA in the database, how that database should be used. You know, there is a public good here, but there's also danger. These are all things that ethicists, bioethicists are grappling with. And as usual, the DNA technology is way ahead of the ethicist in the sense that this technology is growing in leaps and bounds, and the capacity for us to do stuff is greatly eclipsing our ability to understand it at the level of the public. One of the reasons I'm doing these kinds of videos, right, is to hopefully assist people in at least having some idea of what's going on with these types of projects. Okay, where was I? We present evidence that human environmental data is readily detectable from wildlife environmental samples as human bycatch. Demonstrate that identifiable human DNA can be intentionally recovered from human-focused environmental sampling, and discuss the translational and ethical implications of such findings. That's really what they're doing in this particular paper. This is not a revolutionary idea like, oh, we demonstrated that you can find identifiable human DNA in environmental samples. I think every scientist who does environmental samples already recognizes that that's possible. Right? Has been possible for a while, becoming more possible now because our technology is improving. Before, it was like you capture little small pieces of DNA, very short pieces of DNA, which you would have trouble like saying with very definitively this belongs to this particular person. But now we're being able to capture larger fragments of DNA and construct larger larger segments of one's genome. And the more sample you have, the greater your accuracy is at identifying the individual for whom that DNA belongs. Okay, so let's just run on down to the figures. Right? You know, and so they're saying, like, look, you got human activities that release DNA. You are releasing DNA constantly. Just as you walk through your environment, you're sloughing off cells, urination, defecation, right? All that is leaving DNA behind, right? DNA is going into the sewer system, and then it's going out into streams and rivers, right? So if I took a sample from I've been walking around outside in my yard, right? And I have a little stream, a little creek in the back. If I went downstream there, I would probably be able to detect my own DNA in there at some point. I mean, this is a very sensitive technology. I might have to sequence hundreds of millions, hundreds of billions of little fragments, of which most of them are going to be bacteria and, deer and, all kinds of other organisms. But the technology is such that, the machine is going to do all those sequencing, and then I can have a giant database, and then you simply search the database with a human genome. That doesn't even have to be my genome. It just has to be a human genome, right? You sequence with that. You search with that human genome to compare to all those sequences. It's just going to pull out, like, here's all the human sequences, that you sequenced there. And then only if I had my genome, like if I had my actual genome sequenced, right, and I had that available to me, I could compare that to those particular sequences and say, like, oh yeah, there's some particular unique sets of sequences there that are unique to Joel Duff, right? Or they could be my kids too. I mean, it'd be hard to get down to the really specific level of me, but certainly within my family. All right, so what do is you acknowledge that, look, we're all giving off DNA. So the DNA is in the environment just like it is for other organisms. We extract DNA from the environmental samples and recover all the DNA present in that particular environment. And that could be from the soil, it could be from the water, it could be from the air. I have a soil extraction kit in my lab. So you can just take soil, you run a buffer through the soil, And then from that, then you centrifuge basically the soil out. You have this liquid. You take that liquid then, and then you apply, well, basically it's a type of soap that's going to break open any cells. So you can get actual living cells or maybe dead cells that haven't decayed yet, break those open, release the DNA from the nuclei. But the other thing is organisms die, and as they decay, sure, a lot of their DNA is decayed, but they have hundreds of billions of base pairs in hundreds of billions of cells. Some of that DNA escapes into the environment as naked molecules before it's decayed by enzymes. And sure, it's going to be decaying in the environment as well, but because there's so much DNA, there's going to be little fragments that survive, and they're going to get captured by this process. Then you use what's called untargeted shotgun sequencing to recover reads of the organisms. Well, what untargeted shotgun sequencing is methods today where I am not targeting a particular sequence. If I target in one of my labs that I teach, right, students extract DNA from a sample, and then they're interested in a particular gene, and we want to go in and look at that gene and look at the variation in that particular gene. And so that's a targeted search. And they use something called PCR, polymerase chain reaction, in which they have little pieces of DNA that they know the sequence of, and they know they match a particular gene in a particular organism. And that then is the guide that goes in and finds that location and then copies out that particular gene from the genome. And then we just look at that one gene. In this case, what we're interested in is just like all the DNA. Like we're interested in every little bit of DNA, no matter what it comes from, what organism it comes from, what part of the organism comes from, mitochondria, the chloroplast, right? The nucleus, the X chromosome, the Y chromosome, whatever is there just gets sequenced. And that's a newer technique, not new anymore, like 20 years old, really. Let me just show you what I'm talking about, because this is the instrument used in this particular paper. There are lots and lots of different sequencing products out there. But Oxford Nanopore is a company that produces something called the MinION. And they have a bunch of other products too, as you can see. The Grid Ion, the PromethION, all right? But we're looking at the MinION, and this is the exact instrument used for this particular paper. So I'm going to show you they sequenced hundreds of millions of sequences. And they used it using this little device right here called the MinION. This is a portable, you could put this in your pocket, DNA sequencer. Right? It attaches to your laptop. It could attach to the laptop I'm using right now. And so you just have a USB port on it. You attach it to your laptop. The laptop provides the power to run the DNA analysis. Let me scroll down here. That little MinION, is around $1,000, and that includes the chemistry that you're going to need in order to prepare your sample to have it sequenced. All right, so for $1,000, you get this instrument plus the chemistry in order to sequence hundreds of millions of base pairs of sequence. And you can do this anywhere you have a laptop in the world. Right? So you could go to in the forest in Africa, and you could collect sample and you could extract DNA, and there's some crude DNA extraction kits as well that are very simple to use. And in some cases, you don't even have to extract DNA anymore. You can go straight from a sample, environmental sample, to sequencing. Not so much with this kit. But extract some DNA from your environment, put it in, you mix it with these chemicals, put it into this MinION. Oh, let me go down here. Let me just show it to you. Here we go, the MinION. Here it is. All right. Has a little port. You push your sample into that, and the nanopores, little tiny pores on a silicon chip, right? And each little pore, right, uses the electricity, the current from the computer to create a charge, which then pulls the DNA sample through. You're gonna take your double-stranded DNA, separate it into single strands. You're gonna take those single strands and you're gonna pull them through the pore. All right, DNA is negatively charged, so you can pull it through the pore with a positive charge on the other side of the chip. And as it pulls it through the pore, there's a little tiny current that's going across the little pore. And that current interacts with the DNA molecule going by. So there's A, T, C, G, C, G, T. Well, each A, C, T, G is a slightly different chemically, right? And they interact with the charge a little differently, and that creates a little blip, all right? So this thing is measuring the current across the pore as the DNA is flowing by. And it creates a pattern, and the pattern can be read by this computer. Well, this is feeding out the information into your computer. You have software on the computer which looks at the pattern and says, oh, I can see that an A, then a T, then a T, then a T, then an A, then a C, then a G, then a G, then a G went by because I see this particular pattern of changes in the current fluctuations. And the chip has millions of pores, which means at one time, at any moment in time, 1 million different little strands of DNA could be pulled through the pore. And it's pulling through the pore at hundreds of base pairs per second. And so you're reading off hundreds of millions of base pairs, within just a few minutes, and feeding it into your computer. The computer reads the pattern and spits out the sequence and puts it into a database. And so now you have 100 million base pairs, 100 million sequences, and then you just simply search those sequences and ask, what are they, right? Compare those sequences to other known sequences to figure out what you've got there. Right? So as I said, up to 50 billion base pairs, right? This little instrument can sequence up to 50 billion base pairs from a sample. So that's an enormous amount of sequence from just this little tiny instrument. As like I said, this is something that you could teach a high school student to do. They could get DNA from the environment, they could prepare the sample, they could load this thing, and on the computer they could see their samples, and you could write a really simple script for, I just want to know whether there's a certain organism in my environmental sample, and you go out and look for a sequence from that particular organism. Oh, yes, it's there. All right, so incredible technology, right? Really easy to use today by pretty much anybody, with just a small amount of training. Now, to really fully take advantage of this technology and really fully take advantage of the data set that you're going to get from this little device, you probably should have a good background in molecular biology and so forth. But the point is that this is cheap technology now, allowing you to sequence hundreds of millions and billions of base pairs from the environment. And therefore, if I want to know something about all the organisms that have existed at a particular point in time in a particular place, I can find out for a reasonable amount of effort. And this is why you're seeing so many new samples from so many new places. And this is why we can even afford to do it over, I could sequence the same sample from the sewer output in a particular city, every single day, right? Once you have this instrument, you just keep loading up a sample every day, spit out all the sequence, put it into a pie chart that tells you, here's all the different kinds of things, right there. And maybe you're searching for particular diseases, right? Is there, how much poliovirus is in the sample? How much norovirus is in the sample? How much, SARS virus is in the sample, and so forth. And that tells you that individuals in the environment are sloughing off those particular viruses, and you're able to collect them. All right, let's get back to our paper. All right, I got to step it up here a bit because I thought this was just going to be a little short video I was going to make. And I can tell that it's already been a while, and I've got a lot of things I wanted to say about this. Okay, let's go down here. So you get all your DNA. Then you ask questions about it, how much DNA did you find? And so in this case, they're saying we collected data, and these are the sample names down here. So this is a sample name from a dock, inlet, fort, a tank. These are from their own area where they're doing research on green turtles, right? And then they have tidal pool and other places near the shore. And they've collected samples and they've sequenced DNA. And what they're asking here is the number of human allying, here we go, reads per 10 million. So what they found is in some cases at their own facility, they're finding about as much DNA, human DNA in their samples as they were the green turtle DNA, which is their organism that they're studying, along with lots of other things, of course. So number of human-aligned reads per 10 million. So for every 10 million fragments that they sequenced, not individual base pairs, fragments. So 10 million fragments. Fragments might be tens of base pairs to hundreds of thousands of base pairs in length. All right, so you take all those reads, and they have to be long enough to be able to say, I can tell this is human. So probably not 10 base pairs, right? 10 base pairs could be anything. So hundreds of base pairs or more. And you ask how many of them are human. So how many out of a million? Oh, I'm sorry, out of 10 million. Out of 10 million, 40,000 of them in this particular inlet at this fort, 40,000 out of 10 million were human. And you're thinking, well, that doesn't sound like very much, especially when you get down here, less than 10,000 out of 10 million. So a very small percentage of the sequence is human. But remember, they're doing an environmental sample. The environmental sample is going to include a heck of a lot of bacteria, all right? A bunch of, think of all the different things that are living in that particular environment. So how much environmental DNA is there going to be? A whole lot of it. I think it's almost shocking that there's that much human-aligned DNA in those samples. Then over here we have sand. This is where it just go to a beach, and we'll see in a moment that they did this. This was a rough study in which they just looked at just a first study. Then what they did was they went to a beach, which is very pristine, where human beings aren't allowed to go, all right, preserved, versus beaches where humans are found, versus an actual footprints, all right? We're just going to take sand for where somebody stepped. And you, as you could guess, not much DNA at all at a beach where no humans are at. DNA where humans are at the beach, and then more DNA where the footstep is because some of their cells have sloughed off at that particular point. So if you just take sand, so if you just take some sand, take a little scoop of sand, run the solution through it, that pulls out the DNA fragments, right? Extract DNA from that, sequence all that up, and out of 10 million, you still get a couple thousand fragments that you can identify as being from humans. I mean, humans are a large organism in the environment. We're giving off a lot of DNA through a lot of cells that we're sloughing off. So it's not surprising that we're found all over the place. For the sake of time, I'm going to come on down here. And I only roughly skimmed this paper. So let's just take a look at some of these things. So human environmental DNA from intentional sand sampling in Florida. Right, so we went out and we intentionally sampled trying to find humans, right? So we wrote a protocol where we're searching for human DNA out of the environmental sample. And we have beach sand, very little. We have human footprint sand, right? So they took sand where there's human footprints and they tried to extract DNA from that, and they got some of that. And sand from a restricted access island, and there's no real detectable DNA there. Probably just a few, like I think they actually said there was like a couple fragments that were found there, but they're not large enough to show up on this particular sample. This was in the actual volume of DNA found. Now, why are you all wet? You say hi? You in the video? Oh, yeah, you want to be in the video too. Okay. Yeah, you better get down. All right. Okay. Air sample. So this is where they're actually pumping several hundred liters of air, right, from a room through a filter, and then capturing, again, DNA is negatively charged. So if you have a positive charge filter, you can capture the DNA on that filter, and then you can extract the DNA from that filter, sequence that up. And so, they have, all right, we filter samples from where there's no people in the room, at least no people recently in the room, right? Don't find much human DNA in the air, all right? It does fall out of the air, like your cells, all right, are falling out of the air. People in the room for a short period of time, not so much, and eventually people in the room for a longer period of time, and you start to see actual samples that you can collect. This is showing that at a creek site, they found 330,000 human-aligned samples from their run. Right, 330,000. That's individual sequences. Now, this creek was in somewhere in a city in Florida. They don't say where, and for good reason, because they've sampled 300,000 samples of human beings, and then they've analyzed those sequences, and they discover things like they test them for like really broad groupings of ethnicity, and are able to identify at least just a rough snapshot of the composition of people living near that particular or have access to that, or their DNA would have access to that particular waterway. But really the concerning thing is things like, they have enough sequence that they can look at particular genes, particular areas where, like, we know there are markers, genetic markers for different diseases. You'll say that, there's a particular genetic marker here that we found in a sequence that suggests that there's a person here who has a genetic disease. And I think in this case, one of them was an actual genetic disease that doesn't show up till the person's like, it's a neurological disease. It doesn't show up till they're like 40 or more. So here's somebody who sloughed off some of their DNA. It's in the environment. Somebody has picked it up and actually sequenced it and knows that this person, whoever they are, they don't know who they are, right? But this person is going to develop a neurological disease most likely in the future. Do they know about it? Do they want to know about it? Do they not want to know about it? There might be enough sequence that if others donated their sequences, right, to be analyzed, they could connect that sequence to that particular person. Right? So all of this tells you I'm just giving you a rough snapshot that I'm just trying to give you the idea that your DNA is out there, right? It's sequenceable. And large enough amounts, potentially, that a lot can be known about you. Is that DNA coming from a particular area of the genome? No. This little figure just shows all your chromosomes. And then the chromosomes on the outer ridge is the sample from the water sample, because the water best preserves the DNA. There's the highest concentration of DNA. That's where our DNA is most likely going to end up. And all these little spikes up here just show you the amount of sequence from different locations on the chromosomes, right? So they have hundreds of thousands of sequences, and then they ask where they align on the human chromosome, right? Because we all have the same chromosomes and our genes are roughly in the same locations. And you'll see there's little bits and pieces of sequence from every single chromosome. They're not all like, oh, a certain chromosome preserves better in the environment and other chromosomes decay faster. It's just kind of, it seems very random. And this includes this is not showing it here, but the mitochondrial DNA as well, which you have a lot of copies of. And so that's the easiest thing for us to capture is your mitochondrial DNA. I'm going to try to read their language here. Haplotype analysis of human mitochondrial aligned reads for each shotgun sequence. So the mitochondrion, I remember that your mitochondrion tracks your mother's lineage. And we have like basically a breakdown of the entire history of humans. And depending on what particular mutations you have in your mitochondrial genome, you can be traced in terms of your history on the maternal side. And so that's how you can break it down into there are particular mitochondrial genomes that are in African lineages versus Asian versus Native American and so forth. And so here are their pie charts from the sand, from the water, from the room, and so forth. So showing that, yes, we can capture that kind of information. All right, so quick read of the problems and the benefits, right? Because that's kind of what I wanted to do here. Potential problematic implications of the capture of human genomic environmental DNA data. Requirement of human study-related ethical approvals for wildlife studies. I mean, what happens when we start just getting so much human DNA all the time? Who's going to govern that? Is this something that the university needs to govern? Of course, there's private companies doing this too. Who's got the ethics board that considers how you're using these particular samples? Then there are national databases. How do they going to control the flow of information? Right? There's lots of different levels at which people might have influence over how this data should be used or not used. Right? You don't have human subject consent, right? So is there some kind of breach of privacy if somebody's sequencing your DNA? There's public deposition of eDNA that contains human genomic data. Yep, definitely. There's inadvertent location tracking, right? here's the sample, and when I submit samples for environmental DNA, you're saying what the location of that environmental sample was, because that's important information for anyone else who wants to use that DNA as part of a larger study. That's the great thing about these large databases is, oh, I can go in and I can collect data from other people's studies, add something more to it, come up with new information. Inadvertent genome harvesting, collecting entire genomes. If you get large enough sequences and start putting them together, you could reconstruct the genome of an individual. And now with a whole genome, you could even compile a number of different characteristics of that person, right? You could even potentially draw a profile of what that person potentially might look like just from that. Which of course leads to potential malicious applications. Genome harvesting, right? That's harvesting human genomic data for whatever purposes you might want to use it for. Accumulation of human genomic data for commercial purposes, right? Genetic surveillance, individual tracking of people. Bioconspiracy of human genetic data from populations and countries, right? Do I want to know whether, a particular politician has been in a particular place? Do I want to know what their genome sequence is? I mean, all that's already been possible because anything you touch, you're leaving your DNA on. So, especially, that's not really environmental DNA. It kind of is because you're leaving it in the environment. But if you're targeting a particular person, you say, like, I know they just drank out of that cup, then you're trying to get their sequence and know about it. This environmental DNA thing is kind of like, hey, this is out somewhere where I have no idea whether somebody was there or not, but I could go and I could collect DNA and eventually get a lot of samples. And then I could try to figure out who were the people that were there. Or if you're at a crime scene, you could take samples, which they do take samples at crime scenes, and figure out all the people that were there at that particular location, not just right there at that particular, on that particular knife or something. I don't think it takes a huge amount of imagination to think that there's problems with this. But of course, there are also benefits too, right? We can learn a lot more about human genetic variation. This is a way of sampling undersampled populations. Some people sample this way as a way of getting around right consent, right? I can go to a Native American burial ground, right? And you wouldn't want to, dig up the bones. That's created a lot of issues in the past, a lot of ethical issues there. But if I just simply sample some soil, there's going to be some DNA in that soil. I can go to a cave. I could go to a hunting site where there's a hearth, sample some soil around the hearth, right? And because people are sitting there, and they're going to leave some of their DNA there, and then sequence that DNA. This is also a way of capturing DNA from the past, right? Not just the present population, but also historically. And so we have lots of samples of humans from thousands of years ago in this way as well. We can do disease risk susceptibility studies. We're doing that all the time, and that's constantly expanding, right? How much disease is in the environment? You can get right down to very local. I can go to a particular sewer pipe that only serves one street, and from that I can determine how many people on this street are carrying certain diseases. See, that starts to get more personal when you get that close. It's one thing to say like the whole entire city sewer. Now I know that like somebody in the city has this condition, but when you start like narrowing it down, like okay, well, which particular pipe was bringing that? And then I keep going down. And you can keep getting down to the point where like it comes from this house, at this point. And because this technology is becoming cheaper, that's not like, oh, this is going to cost me hundreds of thousands of dollars to do, right? This is becoming more and more feasible because it's not that we haven't been able to do it technologically. It's that the cost has come down to the point where it's becoming like anything you dream up as a potential project to do is becoming feasible. Non-invasive way of monitoring things. I don't actually have to kill an organism or take a feather off. I don't have to cut a little piece off of an animal in order to extract its DNA, right? I just extract it from things it's left behind. You can do continual health monitoring, right? You can continue to sequence yourself, right, by providing samples. forensic and criminal investigation applications, right? We already know about those, and they're just ever expanding. And then the question becomes too, things like, if you're, who controls how that is used? Right now, it just seems in the United States, it's kind of a free-for-all. There's some kind of basic rules about thou shalt not do this, but if there's no one that's written a rule, basically everyone just says, well, I can do whatever I want, if no one said I can't do it. So there's probably going to need to be more regulation there. And of course, in our environment, we have people that don't like any regulation. People want more regulation, the whole debate about regulation. But this can get, who's in control of this information? Really important questions here. Maybe help locate sites of archaeological importance. If you do lots of DNA samples from lots of locations that you think maybe people lived there in the past, like lots of different small caves or something like that, you could do a quick soil sample, right? Or drill down deep into the soil in that particular cave, take a couple samples out there, combine them all together when you're DNA extraction. And if you get some human DNA, and that human DNA looks interesting, right? Then you might say, like, hey, it's worth going to that particular cave and actually doing a dig. We might find some tools. We might find other evidence of people having lived there, right? It becomes an archaeological site, right? Instead of just like, I don't see anything here, but let's just start digging and see what we find, right? This becomes a quick analysis tool for guiding you to that location is not very promising because I couldn't find any human DNA in the soil at this location. So probably humans haven't spent a lot of time here, right? Yeah, so welcome to the wild world of environmental DNA and inadvertent human genomic bycatch in environmental DNA samples. Now, when I did work with micropical plankton out in the Arctic, we were looking for a specific gene. All right, so we were collecting environmental DNA, but we weren't sequencing every little bit of the environmental DNA. We had specific primers that targeted a particular gene that's present in photosynthetic and non-photosynthetic unicellular protist, right? And so those particular primers were specific to a gene that's found in those particular organisms. Actually, they're found in that gene is found in human beings too, but the sequence of the primer we're using doesn't really target humans very well. So I never saw any human sequence in my particular sampling strategy. And even if my own DNA got into that sample, it wouldn't necessarily amplify well, and therefore I wouldn't have sequenced it. But what we're talking about today is I didn't have that mini-IN kit, because the easier thing to do today is instead of doing all the work of targeting a sequence and having to go through, there's a whole bunch of procedure to amplify a particular gene and then clone it, and it's a messy process. This newer process of just like, hey, it's so cheap just to sequence everything. You don't need to target anything. Just sequence it all. Just do it all. Get your 50 billion base pairs and then just use a computer to sort out what they all are. And then, you don't care about the humans because you're interested in something else where you can just like ignore the human DNA. The question is, do you just ignore it, right? Do you just throw it away? But that's part of the environment. So should you submit that as part of the environmental sample? These are all really interesting questions that bioethicists, biologists, molecular biologists, and eventually regulators, politicians, and so forth have to grapple with. Complex systems like this. But research like this that we've just looked at is an important foundational thing just to help us all be aware of just how easy it is for us to leave our DNA around and have it sequenced. All right, that's it for today. Thanks for listening. Bye-bye.